Hi, this is duly noted covering several of the cranial nerves as they move into parts of the orbit and also related to the vestibular apparatus and the cochlea. So in this lab we covered a lot of cranial nerves, so I wanted to review some. I did not include the optic nerve pathway in this one because it will be a separate video because it's more complex. These are relatively simple and we're able to fit on one board. Just a little zoom in to remind you of the key. I tried to keep all the colors consistent on the videos as much as possible since the occipital temporal gyri are responsible not only for shape but also for colors, and you'll remember a little bit better if you use some colors. If you're colorblind, I'm so sorry. Let me point out that this is blue, this is black, this is purple, this is green, and this is pink. Hopefully I've done enough labeling so you can at least find it if you are colorblind. My apologies for that. You can see the dotted pathways are presynaptic and the solid lines are postsynaptic. We have a presynaptic pathway for somatomotor that's dotted in blue. This is general somatic efferent, first order neuron. The solid line postsynaptic somatomotor, GSE, second order neuron. This is going to synapse, of course, on a nucleus because all somatomotor and somatosensory synapse on a nucleus. The somatomotor does not use a ganglion for any purposes. The dotted line you see here is presynaptic somatosensory. This is general somatic afferent, first order neuron. The solid line in black is postsynaptic somatosensory. This is general sensory or general somatic afferent for second order neuron. This uses a ganglion for the presynaptic pathway where it houses the pseudounipolar nerve cell bodies, but there is no synapse present there. It synapses on a nucleus because all sensation, all somatomotor synapse on a nucleus and not a ganglion. The presynaptic pathway for a special sense, this special sense of afference, special somatic afference, this is going to be for both the vestibular apparatus for this breakout and also for the cochlea. So the dotted line you see is presynaptic, special sense. Oh, it's not taste though, my apologies. This is the special sense for vestibular apparatus and for cochlea. So I'll just erase that for you, my apologies. And so it's first order, and then the second order is on a nucleus, which you'll see here is going to be a solid line for the postsynaptic pathway. We're going to use the vestibular and the cochlear nuclei. And so we have SSA, first, second order neurons, but first order is going to be dotted and second order is going to be solid. Now the presynaptic and postsynaptic pathways here for parasympathetic and sympathetic, these are going to be preganglionic and postganglionic. So the synapse does occur at a ganglion. The nerve cell body is going to be located both in a nucleus for pre and then in a ganglion for post. So we see a dotted line for green. Presynaptic parasympathetic is going to be for general visceral efferent. This is first order. The solid line is postsynaptic parasympathetic for general visceral efferent, second order. And then this presynaptic sympathetic and postsynaptic dotted and solid respectively for general visceral efferent, first and second order. So let's get started. Let's trace one pathway. You'll see the structures here that we listed are going to be in brown just to help you differentiate the fact that this is a structure that something is coming to or going from. Let's pick a structure. Let's pick some of these nuclei that go to the extraocular muscles first. The ocular motor nucleus, we can see here, we can see the trochlear nucleus, we can see the abducens nucleus. These structures are going to be our structures that innervate the extraocular muscles. There are 7 of these structures, and we'll cover those in just a second. They start at the precentral gyrus of the frontal lobe, where we take the cortical bulbar tract. Notice that it is a dotted line because we have not synapsed. It's a monosynaptic pathway. Then we take the cortical bulbar tract to our respective nuclei. You can see them going down to respective spots, and so the ocular motor nucleus is going to go to 5 muscles. And so we see a superior division going to the superior rectus and to the levator palpebrae superioris. The inferior division is going to go to the inferior rectus, medial rectus, and the inferior oblique muscles, making 5 muscles innervated by the ocular motor nerve. When we come down over here to the trochlear nucleus, same pathway for presynaptic, we synapse on the nucleus and we send out through our cranial nerve 4, trochlear nerve over to the superior oblique, innervating 1 muscle. We can also go to this presynaptic pathway over to abducens nucleus where we synapse, and our cranial nerve 6 is going to take us to lateral rectus. The way many remember this is LR6, SO4, everything else is 3. for the extraocular muscles. Again, LR6, SO4, everything else is 3. A great mnemonic to help you remember the 7 extraocular muscles and their 3 innervations. On ocular motor travels some preganglionic parasympathetics that we should discuss. From the Edinger-Westphal nucleus, we travel onto the ocular motor nerve, and then we pass from the ocular motor nerve over into the orbit to a ganglion that's suspended from branches of V1 called the ciliary ganglion. Notice there's a solid line here. We synapse on the ciliary ganglion and we head out on branches of V1, particular branches called short ciliary nerves. The short ciliary nerves then take us over to the ciliary muscles that control the shape of the lens and the sphincter pupilla muscles that constrict and decrease the size of the opening of the eye. So for parasympathetic pathways that are basically rest and digest, you don't need a lot of light, so we're limiting light from coming into the eye, and we're accommodating and changing the shape of the lens from anterior to posterior so we can become more nearsighted. A lesion of this would result in mydriasis, wide-iasis, we're letting in too much light, or cycloplegia, where we could not change the shape of the lens and we lose our accommodation. As we come back, we can actually trace out from that same pathway back to the trigeminal nucleus. Remember that nasociliary nerve was suspending the ciliary ganglion, and so we can sense sensation from the ethmoid air cells, from the skin at the medial canthus, to the sclera, iris, and cornea. We're sending things back up to the short ciliary nerves, back to the nasociliary nerve. We travel on the ophthalmic division, which gave rise to the nasociliary nerve, and then go to the trigeminal ganglion. Note the line is dotted because there is no synapse here. It's the house of sooty unipolar nerve cell bodies. We then take from our peripheral process back to our central process, going back in through cranial nerve 5, and then heading to the trigeminal sensory nucleus, where we have our first synapse, because all sensation, all somatomotor synapse on a nucleus, making this a solid line. Traveling down now, we have some really interesting pathways that are for special somatic afferents, and this is going to be for the cochlear and the vestibular apparatus. So here's the cochlea. This is going to be for hearing. As you guys probably already know, we have a sound wave come into the tympanic membrane that hits the malleus, which articulates with the incus, which articulates with the stapes. The stapes pushes into the oval window and helps us to create a wave that goes through the cochlea and then bends a hair cell. When we bend the hair cell, it creates an action potential that then travels through the cochlear nerve, which is part of the vestibular cochlear system. It goes over to its own ganglion called a spiral ganglion, where we have bipolar nerve cell bodies. Again, there's no synapse here. Note the dotted line. We send our central process in through the vestibular cochlear nerve, where we travel into the cochlear nuclei that's located basically at that junction of the pons and the medulla. We have a dorsal and ventral set of cochlear nuclei. Then we travel up the pathway up to these amazing nuclei called the inferior collicular nuclei, and then we can travel over to the brain and be perceived in the transverse temporal gyri of Heschel if we're so inclined to hear this actual signal that came in. For the semicircular canals, you have 3. You have anterior, posterior, and lateral semicircular canals, and we can actually change the shape of our head and change this flow of endolymph and perilymph that then can stimulate an action potential that travels through the vestibular nerve. We have 3 different routes that travel in, and then the vestibular nerve then will go to its own ganglion called the vestibular ganglion. The vestibular ganglion, of course, is bipolar nerve cell bodies for a special sense of afferents. And now we'll take it in through the vestibular cochlear nerve, and then we'll finally get to our synapse, which is at one of 4 vestibular nuclei located at the junction of the pons and the medulla. If we deem it so inclined, we'll take it up to the thalamus and then take it up to the cerebrum to be perceived. Now, for our sympathetics, remember that all sympathetics for the head have virtually the same pathway. We have the lateral horn from T1 to T3, preganglionic sympathetics that travel on ventral rootlets, then go to the ventral root, then to the spinal nerve, then to the white ramus communicans, and then to the sympathetic chain. We then synapse on our superior cervical sympathetic ganglion and travel on the internal carotid plexus, where it gives a branch called the ophthalmic artery that has an ophthalmic plexus that will then head us to particular structures like shutting down our parasympathetics at the ethmoid air cells. Our sclera and cornea don't receive much there, but we do go to the dilator pupilla. The dilator pupilla are what dilate the pupil and counter the sphincter pupillae up top. This will dilate our pupil to be able to let more light in so we can run from our predator. So hopefully you found this breakout very, very useful, and I'll take a screenshot of this and place it into the comment section and also into the readout, so that when you're actually reading what's actually in this group, you'll be able to click on a link and head right to the picture. Thank you so much for watching the video, and hopefully you find it useful.